First off, when you think about science, you're probably thinking the scientific method, but that's just one way of doing science. Okay, it's not the only way, it's not the be all end all, but there are a couple of basic concepts we need to make sure that we understand. First off, theory and law, hypothesis, prediction, sort of those base terms. Theory are mechanistic. They're trying to explain how something occurs, whereas laws are trying to explain what happens under a given set of circumstances. If you have evidence against either one, then they will be not supported, and that's going to be problematic. Hypotheses are going to be based upon sort of relationships between variables, and predictions are going to be directional. So if this happens, then I expect this result, that is a prediction. Okay, however, if I expect there to be a relationship between, I don't know, fertilizer presence and plant growth, that is a hypothesis. Okay, now null and alternative hypothesis are statistical hypotheses. A null hypothesis, there is no relationship, and alternative, there is a relationship. So when you are trying to use, say, chi-square or something, you're trying to support or refute the null hypothesis. You're never trying to prove that. It's not statistically accurate language. When you look at variables, dry mix is a very useful mnemonic, dependent versus independent. Okay, the responding variable versus the manipulated variable. And then you have your controls. Positive controls are going to be a control that is going to give you a positive result for comparatory purposes. Negative control gives you a negative result for comparatory purposes. That is different from controls, okay, or a controlled variable. Those are things that are held constant so that only your manipulated independent variables are being measured. Whereas the control group is going to be something that is not exposed to that variable for maybe baseline comparison or differentiation of results. When they ask you an experimental design based FRQ, it's going to be something like the bees and caffeine one that we did a long time ago. Okay, they're going to give you a great deal of background information, they're going to give you some sort of data, but then the actual question will ask you to design an experiment using whatever and identify specific components. So in this case, it wanted you to identify the null, an appropriate control, and the predicted results. Okay, so the null would be that increasing caffeine would have no effect on the number of floral visits. Okay, the control would be nectar or flowers with no caffeine compared to nectar or flowers with caffeine. And then your predicted results, if there is a relationship there, the number of floral visits by bees will be different at increasing caffeine concentrations would be valid, or the number of visits would be different than the control. So in comparison to the control, there will be a difference. When you start moving into overall data and analysis, a lot of it's going to come down to either representation of data or basic calculations. So you will need to be able to construct basic graphs or charts or that kind of thing, but more often than not, it's either a line graph or a bar graph. If it is categorical things, then you would do a bar graph. If it is a continuous variable, then you would do a line graph most likely. Either way, there will probably be error bars on that graph. Remember that error bars should be set to 2 standard errors, which is equal to 95% confidence. They could also ask you to interpret a wide variety of graphs and plots, and it's not limited to the ones here. They give you some strange graphs sometimes. Just a standard XY graph, a log Y graph, where the log scale is happening so that it's not changing by equal levels. Histograms are going to be looking at bin data and kind of sort of categorical. Dual Y will have 2 different Y axes. The box and whiskers, what you can see an example of here, where you're looking at quartiles and medians. Pie charts, looking at percentages, all of those things are fair game. When you move into calculations, make sure that you are familiar with the formula chart so that all of those things are fair. Rate is going to be a change over time or a derivative. Rise over run, all of those could be applicable. You will need to be able to do quite typically some really basic non-formula chart calculations like percent change, which is just final minus initial divided by initial times 100. or efficiency, which is useful output over input times 100. Other things that they have asked in the past are basic metric conversions or dimensional analysis questions, confidence interval and error bar interpretations. Remember, if the error bars overlap, there is no difference. You could also have to do some sort of hypothesis testing like a chi-square. Okay, any of that is fair game. Now, the chi-square formula is on the formula chart, but you need to remember the difference between the critical variables, or the critical values rather, and the chi-square value. So if your chi-square value is greater than your critical value, then you would reject the null hypothesis. If it is less, then you would fail to reject. And then the other part is, are you going to support or refute the alternative hypothesis? The elements we need to maintain living things exist in varying amounts. You have macronutrients, micronutrients, trace elements, and they exist a wide range of elements needed to support living beings. Of course, carbon is absolutely essential as it forms the basis of all biological macromolecules. Hydrogen and oxygen exist on their own as well as in water. Phosphorus and sulfur and sodium and chlorine and magnesium, all sorts of elements needed to support living beings. Now, some elements are existing naturally within the food that we eat, and this is one way that people can end up with malnutrition if they're not getting the proper elements that they need. Iodine, for example, we may not need in very large amounts, but without it, we have major thyroid problems. Atoms, of course, are these basic building blocks, these elements that we would find on the periodic table. They're composed, of course, of protons, neutrons, and electrons, all of which alter charge and basic properties. The electrons control ion or ionicity. Neutrons are a major component of atomic weight, along with protons, and protons determine the atomic number or the identity of the element. So if you were to alter the number of protons, you would change the atomic number and you would change what the element is. If you were to alter the number of neutrons, you would make an isotope. It will affect the weight, not the charge, and will alter, so carbon-12 and carbon-14, for example. Electrons are negatively charged, found in the electron cloud, and adding or removing electrons creates ions, cations and anions. These atoms exist as the basic building blocks of life, and they are extremely important not just to chemistry, but to biology. When atoms come together to make molecules, the degree of equality in sharing the electrons determines what type of bond and what type of atom you will produce, or what type of molecule you'll produce. An example here is sodium and chlorine. You have a metal and a non-metal that are very different in electronegativity. So you take something that is a solid, that is explosive in water, and chlorine, which is a gas that is toxic, and you create salt, which we use all the time. But that is an extremely unequal sharing of electrons. If you look at something like H2, H2 is a so-called diatomic element. Because the pull of both atoms is equal, you create a bond that is extremely equal. So the degree of equality of electron sharing is extremely important. That equality there, that pull of electrons, is electronegativity. Electronegativity increases as you go up to the right in the periodic table, so things like nitrogen, oxygen, fluorine, chlorine are going to be the highest in electronegativity. And if you look at any two elements, the difference in the electronegativity will show you what type of bond you will make. If the difference is minimal, it will be a nonpolar covalent bond. If the difference is intermediate, polar covalent bond. These are important for biology because covalent bonds will not dissociate in water. Ionic bonds, however, have extreme differences in electronegativity. There's a polarity there. There's partial charges that exist within both polar and ionic things, and these charges affect biology, so they become increasingly important to look at. So since ionic bonds dissociate in water, while ions are essential for bio, their bond is less important. Due to the partial charge of the polar covalent bonds, there are positive and negative ends in these polar covalent molecules. So these charges do affect our body chemistry. One of the most important ways it affects our body chemistry is the existence of hydrogen bonding. Hydrogen bonds individually are very weak, but as you form many of them with any polar covalent molecule, they become collectively quite strong. Nitrogen, oxygen, fluorine, and chlorine are the only four elements that are strong enough to create this hydrogen bonding potential. and chlorine is too heavy to work within the biological structure. So really, if you look for molecules that contain N, O, and F, you will have the potential for hydrogen bonding. Hydrogen bonding allows all of the various properties of water. We know that water is most dense at about 4 degrees Celsius, and it gets less dense in the solid, allowing it ice to float, creating insulation and a variety of factors that are essential to life. The surface tension, the bonds between the water molecules are stronger than the weight of the object being pushing down, so it distributes that weight and creates properties like capillary action, the meniscus of a graduated cylinder. All of that connects to the broader ideas of adhesion and cohesion, with adhesion being the fact that water sticks to things other than water, and cohesion that water molecules stick to each other. So a straw, for example, works through combining these factors, or the evapotranspiration of a tree. Water is a very good solvent, but only for polar things. So saying that water is a universal solvent is of course false. And water has a very high specific heat. It takes a lot of energy to make water change its phase. And that also helps moderate climate. It creates evaporative cooling, allowing organisms to evaporate heat, because that heat goes into breaking the hydrogen bonds, turning your sweat into a gas. And as that gas evaporates, it carries the energy with it. So water and hydrogen bonding are extremely important to maintaining life as we know it. The movement of water, or water potential, becomes extremely important when you're looking at factors like osmosis and diffusion and various components of living organisms that depend upon water. In the tree example here, there's the most water in the soil, there's the least water in the air. So water moves from where there is high water potential to where there is low water potential. They're going to where there is less water. And evapotranspiration depends upon a variety of properties of water, including adhesion and cohesion, to make this possible. In later sections, we'll look in more detail at water potential, but it's something that comes up quite often. Water molecules, the H2O, don't stay H2O. It turns out they're constantly going back and forth between a hydronium state and a hydroxide state, becoming positive and negative charge, moving those hydrogens around. The degree of protons in the water is connected to the idea of acidity. Okay, so when the protons are higher than the hydroxides, it's acidic. So we're going to use the definition of an acid. An acid is anything that donates protons in aqueous solution. So if protons are coming out, it's an acid. If protons are being absorbed or OHs are coming out, it's a base. So understanding that pH is equal to the negative log of H plus is a major factor in understanding pH. Prime example here is if you look at the concentration of H+, if you turn that into scientific notation, we can find out the pH of any given solution. So in the first example, we have a 1, 2, 3, 4, 5, 6, 7. So that's 1 times 10 to the negative 7th. The pH is 7. For the next one, the pH would be 4. The last one is an OH concentration, so 1, 2, 3, 4, 5, 6. The pOH is 1 times 10 to the negative 6. 14, which is our scale, minus 6 tells me that the pH is 8. So if you take those atoms and you take those molecules, then you put them together in biologically stable groups. That's what a functional group is. These are basically the building blocks of living things. These are Legos of biology. There are several groupings of functional groups. These ones are the ones that you need to be able to recognize on site. And don't freak out. It's not that you have to know every little detail. You need to know that hydroxyl groups, because it's adding a polar molecule, an alcohol group on, that makes things polar. A carbonyl group is a structural carbon-oxygen. It doesn't really have many effects, but it will alter polarity a bit depending upon where it's located. Carboxyl groups make things acidic because it can donate protons. Amino groups make things basic because it can accept protons. Sulfhydryl groups are extremely important in the cysteine amino acid because they are what make proteins able to have their tertiary sulfhydryl bridges. Phosphate groups are extremely essential in things like DNA and ATP. Because phosphate groups are able to release energy when in water and we are aqueous, that becomes a very useful energy modifier. Methyl groups can be used to silence genes. They can be combined, like the example on the bottom, by adding a methyl group to estradiol, we go from female hormone to male hormone. So they do have many effects. These functional groups, it's not absolutely essential to remember a massive list of them. If you know the basic properties, you'll be okay. One of the most important ideas in biology, though, is that shape determines functions. By putting these pieces together in different ways, even if you have the exact same chemical formula, you will have a major effect on the function in the body. So a structural isomer is the same formula, but is rearranged in its bonding pattern differently. A cis-trans isomer is all about geometry and angles. The angles are different. And the enantiomers, you have mirror images of each other, handedness, if you will. Your left and your right hand are the same, but different. And that is the enantiomer or stereoisomer. So shape is extremely important. So morphine, for example, the drug, works because it has the same receptor shape as your natural pain-relieving endorphins. And that's also why it's very easy to become addicted. Water is another major player in how we put molecules together. So those functional groups and the shapes and the isomers, they are put together using dehydration synthesis. So you have to remove water to make a bond. And then hydrolysis, you have to add water to break a bond. This is the most basic formation of covalent bonds in biology. It's used in all of the biological macromolecules, but in every type of molecule that it functions, it has its own special name. So sugars, for example, by putting two sugars together, in this example it looks like a glucose and a fructose, of course it's labeled, you put them together to make a sucrose, and since it's a sugar bond, even though it's just a covalent bond formed by dehydration reactions, it's called a glycosidic linkage. So those molecules that can be put together, they're made of all of those different basic building blocks and those elements of life. We find that we have essential nutrients and we have non-essential nutrients. These things are not just food. It's really, if your body can make the molecule, then it's non-essential. If you have to ingest it, it's essential nutrients. So certain vitamins and minerals and fatty acids and amino acids, our body cannot produce and we must eat. For example, vitamin B3 or vitamin C are both nutrients that we have to get from something else, typically plants or fruits and vegetables kind of thing. And if you don't get the right nutrients, you will have some form of malnutrition. It could be something as extreme as marasmus and kwashiorkor, which are both forms of malnutrition. In marasmus, it's the child on the left. The child is getting plenty of calories, but they're not getting protein. They're just getting sugars and things like corn and rice, leading to fatty liver disease and malnutrition, whereas the child on the right has a complete lack of calories and has kwashiorkor, which is extremely damaging to the body. Both of them are. So those basic building blocks of life really depend upon the functionality and the tetravalency of carbon. Because carbon is so flexible in the way it can bond, it forms the basis of all of the molecules of life. And we get those carbon molecules from things that were fixed by photosynthesis. The enzyme Rubisco takes that CO2, that inorganic CO2, and fixes it into organic carbon that is useful. And that is useful in forming those major biological building blocks that Schnapps refers to, be they proteins, lipids, carbohydrates, or nucleic acids. Carbohydrates, obviously, absolutely essential for short-term energy storage in animals, and they're a major structural component of plants. They can come in linear forms or ring forms. Monosaccharides is going to be the base monomer. I can put two monosaccharides with a glycosidic linkage to create a disaccharide. Those disaccharides, in turn, can be assembled into a wide variety of things. So carbohydrates are very important for energy production, but when you put them into large groups or polysaccharides, they have become much more flexible. Those polysaccharides, starch, cellulose, and glycogen are the three most common examples here. All three of these are made of glucose, bonded in different patterns. If you look at the angles, if you look at the isomers, the angles are all the same in the alpha glucose, but in the beta glucose, the angles alternate. Because the bonds are in the same direction in the alpha glucose and things like starch and glycogen, they are digestible and buildable. If you look at cellulose, because it is beta linked, it cannot be digested by our stomachs, and we need specialized bacteria basically that exist within, say, the stomachs of a termite or the stomachs of a cow to break that down. The other two polysaccharides are exceptions. These are ones that contain nitrogen, and they are used in the cell walls of a variety of organisms. So plants use cellulose as a structural polysaccharide to make their cell wall, but fungus and insects' exoskeletons are made of chitin, and the peptidoglycan is used in bacterial cell walls. The archaeans have a variety of sugars, not one that you have to know. Lipids are biological macromolecules. They are important for things like long-term energy storage and padding and cell membrane production. Certain vitamins, certain hormones are all made of lipid, but lipids are not polymers. They don't have one component that repeats. It's a variety of components that put together into different ways, different things. So oils and waxes and fats, and they're all very different because they're made in different ways. The phospholipid is one of the most common examples of a fat, and this is a modified triacylglycerol or triglyceride that has both a polar and a nonpolar region. Because it is both polar and nonpolar, it's ideal for producing things like cell membranes, with the polar region being hydrophilic and the nonpolar region being hydrophobic, creating an amphipathic molecule that is great for a variety of functions. Triglycerides, and this one's labeled here with the ester linkage, which is the name of the covalent bond in lipids. They are the major molecule of fat, and they come in a variety of shapes and sizes based primarily on how much hydrogen is in the molecule. So if there are double bonds or triple bonds in the carbon, they're said to be unsaturated, and that creates bends in the molecules. The more bends in the molecules, the more likely the fat to be is a liquid. So vegetable oil, for example, is a polyunsaturated fat. If I hydrogenate and force a bunch of extra hydrogens into place, I can take that vegetable oil and turn it into a solid like margarine. So healthier fats are typically unsaturated because they don't stick in the body, where the saturated fats do tend to stick. When you hydrogenate, sometimes you can make isomers that are not as good, trans fats. And so those have been banned in most countries now because of how harmful they are to the body. Proteins, of course, are one of the major building blocks of living things. The amino acids, there's a variety of them, between 20 and 22, depending upon which chart you're looking at. Some of them might all go up to 24. Most of them are non-essential. There's only 8 or 9 that are absolutely essential for a human diet, but all of them are exactly the same. You have this amino group, you have this carboxyl group, and you have the R group all bonded to a central alpha carbon. The amino group makes it basic, the carboxyl group makes it acid. The dehydration synthesis needs to come off of the OH group of the carboxyl and the H of the next amino, and it's called a peptide bond. The R group is different from every amino acid. The NCC backbone is not. So biological macromolecules, proteins are the first one of these that we've talked about that have directionality. You have the N terminal and the C terminal. So you can only add new amino acids to the C end, the carboxyl end. Those amino acids obviously differ in their R groups. You should be able to recognize 2 of these on sight. The one with sulfur bonded in the middle is methionine. That is the amino acid that starts all protein structures. And then the one on the right is cysteine with that sulfhydryl group. And we'll need to see that in the 3rd level of structure in just a minute. But proteins are essential. They make up our enzymes, hormones, storage proteins like egg, motor proteins like your muscles, antibodies, transport proteins, receptor proteins, structural proteins like collagen. Proteins are the major building blocks of animals, and so we need to pay a lot of attention to those. Now, proteins are also having multiple levels of structure. The primary structure is just the covalent peptide bonds between all of the amino acids, that NCC, NCC, NCC pattern that you see. Those then bond with the other backbone components using hydrogen bonding in either an alpha helix or a beta pleated sheet. I always remember that there were 2 shapes for the 2nd level of structure. The 3rd level of structure is arguably the most important. It can be held together by a wide variety of structures. The cysteines coming together to make a disulfide bridge, the hydrogen bonds, hydrophobic interactions, ionic bonds, all of these hold the 3rd level of structure together. The 3rd level is the first one that's 3-dimensional. Ionic bonds are likely to form between charged amino acids. Hydrophobics are always going to fold in because they're afraid of water. So we can see a lot of these structures which will help determine the 3D shape of the protein. And if you put multiple 3D shapes together, you make the quaternary structure. So hemoglobin, for example, is made of 4 proteins, 4 tertiary proteins, or something like collagen is made of 3. So different amino acids come together to make different proteins, and then those are then folded and structured into the larger tertiary and quaternary structures. So shape is exceedingly important, and if you alter shapes, you are denaturing that protein. That's typically something to do with heat or salt or acidity or physical agitation, like whisking, for example. If you're trying to whisk egg whites, that would also denature. So being able to make proteins do their jobs and altering proteins is extremely important. We find if proteins have misfolded, they can cause major damage to the body. So it's not just that they can be non-functional, they can actually be pathogenic. So things like Alzheimer's and scrapie and mad cow disease and all of those various prion diseases, those are proteins that folded wrong and became a major problem. We do have other structures, enzymes that will help renature proteins that have been denatured to a certain degree, but those are exceedingly rare. And then we have other structures like chaperonin, which is a protein complex that allows other proteins to fold properly. So your body spends a lot of effort on folding proteins correctly. If the protein is not folded correctly, it's going to likely be destroyed by something with ubiquitin and some of the various enzymes in the cell that prevent these pathogenic proteins as much as possible. Sickle cell disease is a great example of why protein folding is extremely important. If you change the amino acids, if you have the mutation, so long as the properties are the same, the effects may be minimal. But in this example, we took a polar amino acid at position 6 and replaced it with a non-polar amino acid. That causes the molecule to fold in, that creates that abnormal shape, which leads to the abnormal function of the cell. Nucleic acids are the last of the 4 major biological macromolecules. There are 2 major types of nucleic acid, DNA and RNA, and you can really separate those by the type of sugar they use and the types of bases that they use. Deoxyribose has 1 less oxygen, so that forms the D part of DNA, whereas ribose has that extra oxygen. The purines, A and G, are pure silver. The pyrimidines are cut, C-U-T. You cut the blocks of the pyramid. Uracil only exists in RNA along with ribose, and thymine only exists in DNA along with deoxyribose. Technically, DNA is also typically double-stranded and RNA is typically single-stranded, but that last one does have exceptions. You should be able to recognize the purines and the pyrimidines, not by their molecular structure, but at least by their letters. And we know from Chargaff's rules that adenine bonds to thymine and guanine binds to cytosine. But if you are in an RNA, then adenine binds to uracil, and we need to remember that. Like proteins, the nucleic acids, the nucleotides, have directionality. Based upon the number of the carbon in the sugar, you have the 3' and the 5' end. The 1' is where the nitrogenous base binds, and you work clockwise around the sugar. The 5' is going to be where the phosphate groups are bound, so that's going to be the energy source, and the 3' is going to be an open OH group for binding. So nucleic acids are going to have directionality, 5 to 3, and they will only grow from the 5 to 3 direction. So if proteins grew from N to C, then nucleic acids will grow from 5 to 3. Now, because it grows in 5 to 3 and DNA is anti-parallel because the opposite sides are mirror images, 5 to 3, 3 to 5, then the DNA will be read from 3 to 5 and will be built from 5 to 3. That directionality becomes extremely important when you get to later units. DNA also is an example of something that has secondary levels of structure. The double helix is an example of a secondary level of structure. That's pretty much the major one for DNA, where RNA and the many different types of RNA have many different levels of structure and how they fold, each one folding differently.